Good afternoon, everyone, and welcome. My name is Ed Schlesinger, and I'm the Benjamin T. Rome Dean of the Whiting School of Engineering, and it is my great pleasure to welcome you to the installation of Jordan Green as the Herschel L. Seder Professor in Biomedical Engineering. The installation ceremony of an endowed professorship is a particularly wonderful moment in the life of an institution. It allows us to recognize the generosity of our friends as well as the distinguished career and great personal devotion of a particular faculty member. Thank you all for being here today to celebrate this special occasion, and I would like to offer special welcome to today's honoree, Jordan Green, his wife Stephanie, their children, Kaya, Luna, and Sid, and Jordan's father, Henry. Bob Seder, son of the late Herschel Seder, as well as all of their friends and family for being here as well. To help in the celebration, I'm thrilled that Ted DeWeese, Dean of the School of Medicine, joins me today. This professorship was established in the Whiting School of Engineering, supporting faculty in the Biomedical Engineering Department, a joint department across the Whiting School and School of Medicine that allows Johns Hopkins to remain a leader in biomedical research and education across both engineering and medicine. We're also very honored that President Daniels is here to join us today to help with this important celebration and recognition. Now, Jordan, a few people are going to speak on your behalf today, but let me be the first to officially say congratulations on this well-deserved honor. You have most definitely made an indelible mark on the world, and we are proud to have you as a member of the Johns Hopkins University and Medicine family. And to you, Bob, thank you. We are so grateful to your and your family's generosity and commitment to the university over many years, and especially in this moment. And today, we honor your family for creating the Herschel L. Seder Professorship. It is now my pleasure to welcome Dean Ted DeWeese to the podium. Ted.
Ed, thank you very much. Good afternoon, everyone. It's a real pleasure to be with you today, and on behalf of the entire School of Medicine and, of course, our partners in the the School of Engineering to really celebrate the newest Herschel Seder Endowed Professor in Biomedical Engineering, you, Dr. Jordan Green. Congratulations, Jordan. And I should have added most worthy recipient of this, because you are, Jordan. Congratulations to you. Obviously to Bob, to your entire family, thank you for the foresight, particularly for your parents, of establishing such an important endowment. These things are extremely important. They allow us to continue the innovation and the reputation that is so well associated with this great university, but in particular, the most prestigious Department of Biomedical Engineering in the world, and allows us to recruit and retain the folks that we need to maintain that and let them do what they do, change lives, people around the world like Jordan. Thank you, Jordan. Bob, thank you very much. Endowed professorships are indeed the highest honor that the university can bestow on one of our faculty members. They allow us, as I said, to recruit and retain the most creative people, make sure that they can stay focused on what it is we do, change lives, improve the human condition across all the things that we do. And I know, Jordan, you're perfectly aligned with that mission and vision, so thank you for that. Obviously, they're very beautiful in many ways, for two reasons, at least to me. One, they provide the opportunity for stability and flexibility a little bit, and therefore creativity, because the stability piece that all of the faculty love is, hey, I don't have to actually worry about generating all of my salary. That's stability. But then because of that, they can start to focus on the work that matters and being a bit more creative, a bit more flexible in what they do, how they do it. And that allows us to then, of course, continue the virtuous cycle of not just the research, not just the clinical care for those who provide clinical care, but that virtuous cycle of training the next generation of leaders. The second piece of why it's so important to me is that they last, because well beyond any of one in this room sitting in this room, or being alive even, this will continue. the Seder Professorship in Biomedical Engineering will continue. And that is so important to society at large. As Ed mentioned, I think this is particularly special because it does bind together, and Bob and I were talking about that a moment ago. I think in some special ways, this professorship in particular, but professorships across the School of Medicine and Engineering, to keep us aligned with what we do well. And sitting right at the crossroads of this is the Department of Biomedical Engineering. And that is a real, very special thing that, Bob, that your family has helped to create. I'm very biased about it, but like any dean of the School of Medicine at any time in history at Johns Hopkins would say, this is the golden age of medicine and biomedicine. They were wrong. Today is, actually, right now is the golden age. Because if you think about what kind of research can be generated, the massive computing power that lets us generate, and all the data that we get to generate out of patient records, out of precision medicine platforms, precision measurement tools, the massive data, and then the ability to process, store, retrieve, and then find interesting patterns in those, the tools that can get generated to create new therapies, new surgical techniques, new interventions in some way. Biomedical engineering sits right at the nexus of all of that. And that's why, again, this is so important. And as I said the other evening, plus the faculty in biomedical engineering are sort of the coolest faculty, except for the plastic surgeons who are way cooler. But you guys are cool faculty, nonetheless. No offense, Jordan, but not quite plastic surgery cool, but nonetheless cool. And the strong partnership, as I noted, between the School of Medicine and Engineering allows us to continue the bold ambitions that Ed and I have thought about and pursue together. So definitely, Bob, to you, to your entire family, thank you for helping to solidify those bonds. I was speaking again to Bob right beforehand, and Bob's father, Herschel, was indeed a graduate of this fine university, 1939. Had to check that, in a degree in economics, and then went on to lead the Milwaukee Valve Company as its president and its chair for many, many years. I think Bob said 50, something like that, years. And he gave back to this institution, his alma mater, in multiple ways, including the painful one of serving as a trustee for more than 20 years, and advised the Whiting School of Engineering, the School of Advanced International Studies, SICE, and of course our Applied Physics Laboratory. And I think, as Bob noted too, I think he had the vision of picking and betting on a winner. And I think biomedical engineering was a great winner on which to place his bet. And Herschel and his wife Ruth were generous supporters of Hopkins in so many ways, including this professorship. And while they're no longer with us, unfortunately, we are here today to celebrate that legacy. And I told Bob, these things, some people think they're a bit trite to talk about things. I think there isn't anything more important than to think about our history and the history of your family, Bob, in helping to solidify this endowment and be able to support somebody like Jordan, one of our best and brightest faculty members. That is why it matters. So congratulations, Jordan. Bob, congratulations to you and your family. It means so much to us. And with that, I'll turn it over to President Daniels.
Thank you, Ted. It truly is an honor and a privilege to mark the continuation of the Herschel L. Seder Professorship in Biomedical Engineering at Johns Hopkins University with the installation of its third chair holder. It sounds like furniture, but you're so much more than that, Jordan. It's great to have the Seders here, of course, a true Hopkins family spanning 3 generations. And really delighted, Bob, that you're here, former trustee who spent a lot of time in this room, and even after being on the board, has given lots of advice and counsel to me over the years, and friendship. So it's great to have you here, Bob. And of course, it's a family affair, Jordan, to have your whole family here, and I know it's great have your wife and kids, but a particular thrill, of course, to have your father here join you in this really auspicious moment. So, Henry, welcome, and I'm glad you could be here in this very important moment in the life of Hopkins, but in Jordan's life as well. So I'll just say it right at the outset, we're really thrilled to be able to honor a fellow Canadian. And I want you to know, Jordan, that our shared affection and devotion to Tim Hortons Donuts in no way was the tipping point in getting you to this moment at the university, but it helped. When Herschel received one of his several Lifetime Achievement Awards here at Hopkins, he noted that too many engineers are content to add or equal to job specifications. There is, Herschel said, no such thing as or equal. For the non-engineers among us, Herschel knew that each problem required a deep commitment to gathering data, understanding the particulars of a problem, and extrapolating a solution that met the moment, met the challenge, or what he might call a one-size-fits-one solution. This ethos, as you heard, is what made Herschel and his Milwaukee Valve Company international success, keeping our homes running, fueling the international oil delivery supply chain, and powering Navy ships across seas and oceans. Herschel's ethos also happened, of course, to help transform Hopkins. When Herschel was a trustee on the advisory committee for our Whiting School, the field of biomedical engineering was in its infancy. But Herschel he saw clearly that the nation soon would need needed to be able to bridge the gaps between our medical clinicians, researchers, and engineers to spur new ideas to change and improve lives. With his urging and support, we were among the first to establish a Department of Biomedical Engineering, and the genius of this department, of course, is that it spans 2 schools of the university, something that others feared to tread, but we did it, and the rewards have been magnificent and enduring from that moment. Now, of course, with support from Herschel's sons, John and Bob, and his daughter-in-law, Deb, we are among the world leaders in the field. I would say we are the world leader in the field of biomedical engineering. And Bob, who's with us today, clearly has embodied this spirit of pursuing unique solutions to critical problems. As head of vaccine development at the NIH, Bob helped to lead preclinical development of the Moderna COVID vaccine. He's also spearheaded the first human clinical trial showing monoclonal antibodies derived from a unique parent cell can prevent malaria infection. And this is something that Bob has dreamed about, has been enthusiastic about, imagined many years ago. And Bob, we're just so delighted that it's coming to fruition and will have great impact, we hope, on patient populations throughout the world. His work demonstrates that even the biggest discoveries reshaping global health can hinge on the ability to understand a single cell. And that's something, of course, that Jordan knows all too well. You're going to hear more about this from others, real experts, unlike a lawyer. But Jordan understands there is no such thing as or equal. Working at the forefront of personalized medicine, he aims to deliver novel instructions and treatment to highly specific types of cells. Jordan and his team are developing NanoBioTechnologies that you'll hear about more in a moment that can command cancer cells to self-destruct, reprogram cells at the root of MS, reverse age-related macular degeneration, extraordinary innovations with the potential to revolutionize healthcare. Every study, every innovation, every breakthrough in Jordan's work reflected in over 80 patents that he has either received or pending, focuses on remedying the underlying causes of potentially fatal diseases. In this, Jordan, your work is a perfect expression of the Seder vision and dedication to finding one-size-fits-one solutions time and time again. We're so proud that you'll be holding this chair, and on behalf of the university, we wish you our heartiest congratulations. To Bob and his entire family, thanks for doing all that you do at Johns Hopkins and for entrusting us to carry on your father's legacy in this way. With that said, I'll now invite Dean Ed back to the podium to introduce Bob and share how he is continuing Herschel's legacy. Ed, over to you.
Thank you, President Daniels. As you heard, it's now my pleasure to welcome to the podium Bob Seder to provide remarks on behalf of the Seder family. Bob is the son of the late Herschel L. Seder. He received his BA from the Krieger School in Natural Science at Johns Hopkins University in 1981, his MD at Tufts University in 1986, and completed his residency in internal medicine at New York Hospital Cornell Medical Center. He, like his father, has served as a university trustee and is a current member of the Biomedical Engineering External Advisory Board. He is currently chief of the cellular immunology section in the Vaccine Research Center in the National Institute of Allergy and Infectious Diseases at the National Institutes of Health. His work focuses on the cellular and molecular mechanisms by which vaccines and adjuvants mediate protective immunity in mouse and non-human primate models of HIV, malaria, tuberculosis, and cancer. His work has demonstrated the importance of the quality of T-cell responses in mediating protection against various infections and the importance of the route of vaccination in generating tissue-resident T-cells for protection against malaria and tuberculosis. In addition, as you heard, Bob has helped lead the preclinical development of the Moderna mRNA vaccine against COVID-19. Bob and his wife, Debbie, support a variety of initiatives at the Johns Hopkins University, including the William R. Brody Faculty Award, the Murray B. Sachs Professorship, the Ruth and Herschel Seder Scholarship, and the Andrew and Dolores Bozzelli Scholarship at the Whiting School of Engineering. We are truly grateful to Herschel and the Seder family for all they have done for the Whiting School, medicine, and the university. Bob, I invite you to come to the podium to say a few words.
So thank you very much, Ed, for those generous comments. Being in Washington, you always start with acknowledging the president. So out of protocol, I just do want to acknowledge President Daniels, who I sat in this room when he started, how long ago? 15 years ago, and came with a vision for the university and for Baltimore. And I think Ron is probably going to be, aside from Daniel Coit-Gilman, the most transformational president Hopkins has ever had, and is extraordinary. And in this difficult time in academia, I think we're all grateful that we have someone like Ron at the helm. So my greatest respect for you, and also for my personal respect for you as well. I'll also say to Dean Schlesinger, who I've gotten to know well over many years, has also really put the engineering department and has made it not a world-class engineering department, it always was, but now he's really moving it aggressively into one of the premier departments in the world. And I think he has a plan with some new funding to really build this out. And I think that will not only solidify the Department of Engineering, but the Department of Biomedical Engineering. And then when I talked to Dean DeWeese, when I said hello to him, I said, congratulations, I think, on your new position. but I think the university's in good hands. So to give you some background, my father had three passions in life: his family, his business, and Hopkins. And that's all he talked about. And since all my siblings went into the family business, I can tell you that Hopkins was the least contentious of the three. And he absolutely loved it. It was, for him, he worked on a factory, he ran a business on a factory floor every day in the south side of Milwaukee. But when he came to Hopkins, it opened the world to him in terms of the brilliance of the faculty. And so he would come home and he would go through it, whether it was from science or if it was from APL or from engineering. But it was just, that was what he saw as the world of how Hopkins, and if you put up the screen and look what Daniel Coit-Gilman said, Hopkins would lead the world. And really that was in his blood. So the history of the chair is quite interesting and begins with Mike Bloomberg. So my father, having manufactured valves could be used in buildings and in laboratories. So as a form of a donation, he was willing to give valves to all the buildings. And so Mike Bloomberg in the 90s was running a campaign to raise funds, and he said to Morris Offit, who was a fellow trustee and a close friend of my father, he said, "We've got enough valves. We don't need any more valves, but what we do need is cash." And so my dad was a little disgruntled, and I can tell you it didn't go down as what I'm saying in a kind of polite company, but my father said, okay, we're gonna make a significant gift. And my father was on the board of the APL, SICE, and engineering, but he picked a winner, and he knew BME was a winner. And so it was clear that that was where the chair was gonna be funded. So that now leads us to the first invocation of the chair, which occurred many, many years ago at the Hopkins Club with Mike Miller. So Mike gets up, and we're all there, and he shows a slide of Isaac Newton, and he says, Isaac Newton in 1663, there is a chair for Newton that persists, and Dean DeWeese mentioned this, that still exists today. Hawking had it, but I think Mike was using it to say after 300 or 400 years that that chair, look at the impact that chair had at Cambridge and over time. Now, of course, those of us in the group were trying to figure out, was Mike equating himself with Newton? But I must say that Mike was extraordinary, and as in that chair, I think he exemplified all of the things that my father really admired, and that was brilliance. He was a great engineer, he was a great scientist, he was a great leader, and Mike had a special way of respecting and treating his colleagues. And whenever I email Mike and say I need somebody so-and-so, the most special thing for Mike was of talking about people like Jordan. And so Mike really was extraordinarily, and what in Yiddish would be called the mensch. So that's the transition to Jordan. So I want to read you, I ran ChatGPT on Copilot. And so when you put in mensch, and I'm trying to screw up the algorithms on Copilot with a lot of Yiddish, you would say mensch is a Yiddish word, describes a person of integrity and honor. It's used to refer to someone who embodies admirable qualities and acts with nobility. Mensch is someone you can admire and emulate, characterized by their uprightness, responsibility, and dignity. In essence, being a mensch, having a strong sense of what is right and decent. It's a high compliment emphasizing the rarity and value of these qualities. And that's Jordan, was Mike, and that's Jordan. And so I think Jordan is really, I kind of knew Jordan would be the recipient of the chair, so it wasn't really a surprise. So I could tell they're calling me, but I was thrilled. I mean, there just could have been nobody in the entire department that I would want to have the chair more than Jordan. And Jordan has those qualities of scholarship, leadership, entrepreneurship, and collegiality. I attended several BME meetings, and it was clear that Jordan was a leader, and he helped craft BME 2.0, which was the curriculum which was impossible. I don't know how anybody can take it, actually, but it was the curriculum that would guide the next generation. No, I mean, it was 190 credits, but but it was clear that Jordan had a vision for the department, not only in his own work, but in training the next generation. So the other interesting thing is I work on peptide nanoparticle delivery approaches. So we actually share common scientific interests, and students that I had trained years ago actually have worked very closely with Jordan. So I actually know his work. So I know the quality of his work, and more importantly, I know the respect he has in the field. So there could not be a better choice for the Seder Chair. We're all thrilled, and congratulations.
Thank you, Bob. Next, to say a few words about Jordan's impact on the Department of Biomedical Engineering is Michael Miller, the Bessie Darling Massey Professor and Director of Biomedical Engineering at Johns Hopkins. And as you heard, Mike was the first holder of the Seder Chair.
Thank you, Dean.
What?
President. Thank you, Bobby. The Seder family has become synonymous with biomedical engineering. Bobby, you took me off script. And so I just want to correct one thing. Everyone knows it's not my style to ever compare myself to anyone like Newton. What I pointed out for historical relevance was that Newton held the Lucasian chair, but he held the Lucasian chair at an amazing place like the Department of Biomedical Engineering. And I said to Herschel and Herschel's sister and his grandchildren and his family, who were all there, that what's going to be most remarkable is to see who will be the subsequent chair holders, as in the case of the Lucasian chair was held by Lucasian, and then Newton was the second chair holder. And I think this is exemplified by Jordan today to see what's going to happen here. And so I'm so delighted, Jordan. Now, Henry and I prepared for this, and I told Henry that I really wanted to do something I don't normally do, which is really try to talk publicly about the serious excellence of my faculty. Of course, I talk publicly about our faculty and how great they are, but I would try to be serious about it and scholarly. And that Henry said that he would do the family side. And so I think together we're going to get both aspects. And as you heard from Bobby, Herschel loves his family, and he loved my family. And Eliza was 2 years old then, and there's a picture of Herschel and Herschel's sister who came to my house for dinner to be with Eliza. And it was a very lovely thing. So now I will turn to my serious statement here of why Jordan should be the third holder. Dr. Jordan Green is the Vice Chair for Research and Translation of Biomedical Engineering and is an expert in immunoengineering and cell and tissue engineering. He works at the interdisciplinary space of biology, chemistry, and engineering, something we talk about all the time at Johns Hopkins, the interdisciplinary space, and he uses nanoparticles to target and control the delivery of nucleic acids and genetic medicines. Nucleic acids and genetic medicines, and how important is that now today? I mean, it's quite remarkable. Dr. Green's lab is pioneering innovative technologies that can directly benefit human health. That's the privilege we have in biomedical engineering at Johns Hopkins. That is a remarkable privilege, to impact human health. Now, I wanted to say a couple examples because I think they're amazing. So he's developing treatments that selectively kill cancer cells, new technologies to enhance drug delivery efficiency, and the design of biomimetic materials to activate the immune system, obviously for tissue repair and other things that we know the immune system does. Now there's, I think, a beautiful intersection with Dean Schlesinger in that Jordan knows I really respect Carnegie Mellon. I think Carnegie Mellon is an amazing engineering school and one that we can emulate. The students engineer at Carnegie Mellon. You see the engineering. Dr. Green received his BS in chemical engineering and biomedical engineering from Carnegie Mellon University in 2003. So I think, Dean, Ed, you would have been the chair of electrical engineering then in 2003. Were you the chair about it? Yeah, but you weren't yet the chair. And he completed his PhD in 2007 in biological engineering at MIT. And interestingly, we, of course, have the name biomedical engineering. MIT is, of course, biological engineering because they don't have the advantage that we have, which is the School of Medicine, our tight integration. And so that's really important. Now, Dr. Green is the ultimate poster child of translation, of impacting the world. Of course, he founded Asclepics Therapeutics, which is now in clinical trials. And he's doing, as Bobby said, peptide-based treatment for age-related macular degeneration. And he's on the board or has founded 5 other companies. He's won numerous awards. He's a fellow of the National Academy of Inventors, the Biomedical Engineering Society, the American Institute for Medical and Biological Engineering. He's an editor of Science Advances. His work has resulted in almost 200 publications, but he holds or is in process order 86 at this time patents, 86 patents. That's remarkable. He's received numerous awards, the Presidential Early Career Award for Scientists and Engineers, Popular Science's Brilliant 10, and was a National Academy of Medicine Emerging Leader in Health and Medicine. Let me finish with a final factoid that Herschel and Bobby and the Seder family will love. I went to look up how many Siebel scholars you have mentored. As graduate students, the greatest award you can receive in the United States is to be a Siebel Scholar. We visited Ron Daniels with several of our Siebel Scholars. Jordan has had 8 Siebel Scholars. That was our count. Now, I may have missed a few in the last 10 years, 8 Siebel Scholars. That's really, really great, Jordan. So with that, I'm going to turn over to my colleague, Henry Brem, to talk more about family and things. Henry, thank you.
Hi, I'm Henry Brem. I'm the Harvey Cushing Professor of Neurosurgery, and I learned already about Jordan, which I didn't know. Jordan is the professor of neurosurgery at Johns Hopkins University. I had no idea you were associated with biomedical engineering as well, so that's amazing. I did want to sort of very personally tell a little bit about my own story and why it relates to Jordan and why, I feel so privileged that he asked me to say a few words. I started really the most foremost experience of my early career was working with an incredible surgeon scientist, Judah Folkman, at Harvard Medical School, and working on angiogenesis. And he showed how a surgeon can also be a scientist and really change the world through that. And when we were working together on the first inhibitor of angiogenesis, a graduate student, a postdoctoral graduate student, came to work on my project under Dr. Folkman. That was Bob Langer. And that led to a lifetime of collaborations between myself and Bob Langer, which led to the first angiogenesis inhibitor and then also the polymers that were used for drug delivery. And that came out of that early work. I went on to, finish medical school, do residency, train to be a neurosurgeon. And when I came to Hopkins, I reignited that collaboration from my student time with Bob, and that led to our developing here at Hopkins with our engineering colleagues at MIT, the first new treatment for brain tumors using controlled release polymers in the, eventually in the 80s, 90s. So I was thrilled. So this whole thing of friendships and relationships is so critically important. And so in 2007, I was thrilled that my brand new son-in-law, Joel Sunshine, took a job to work for a year with Bob Langer, my friend who was a graduate student postdoc with me in the work in the Langer lab at MIT. And sure enough, his mentor there that he was assigned to and began working with was Jordan Green at MIT. And he worked with him as a graduate student and then as a postdoctoral fellow there. And they had a spectacular year and they published very important work and it was a great experience. And I felt like, wow, that's the connection, from my past with Joel and now with one of Bob Langer's trainees. And I did speak to Bob actually on my way over here, and I told him I was going to make a few remarks. And he said to just pass on that Jordan was one of the most phenomenal students that he's ever had, and also one of the nicest human beings. And I think, we'll come back to that. And that one of the, that it was such a beautiful combination of being such a mensch. Dr. Seder, I agree, and a gentleman as well as a great scientist. So Jordan stayed at MIT. Joel came here as a medical student, and then Jordan called and said, he accepted a position in biomedical engineering. I think he started in biomedical engineering. It took a little while to come into neurosurgery. And Joel was all excited, and he started helping him unpack his lab and starting the lab. And Jordan, in his typical mentorship way, said, listen, you spent a year together, published paper, did this whole, why don't you just get a PhD? Okay, keep working, in my laboratory and we'll work together. And so that led to another collaboration. And it's just thrilling for me to see from generation to generation how Joel is on the faculty now in dermatology and in biomedical engineering, and he's mentored by Jordan, and it's one of many, many people that Jordan has touched, not only as a scientist, but as a friend. And I'd say their families are together, children grow up together. It's a beautiful, beautiful thing. And Jordan has affected and impacted many people in neurosurgery and done beautiful work for brain tumors as well as in ophthalmology and so many other areas. And I think that, Dr. Seder, what you said resonates, and what your father felt. If the things that are most important to Jordan are his family, his science, his work, and Johns Hopkins. And we are so privileged at Hopkins to have Jordan as a colleague, as a friend, and as a mentor to the next generation of great scientists and physicians. So with that, I just am very honored to invite you to the podium to give your remarks as the Seder Professor of Biomedical Engineering and Neurosurgery.
Wow, well, thanks so much, Everett. And I'm almost speechless here. As such kind words from President Daniels, Dean Schlesinger, Dean DeWitt, from Mike Miller and Henry Brem. And I feel so lucky and privileged from the first day I've been at Hopkins. It was something as a student and then a PhD postdoc, interviewed different places, and I remember talking to Bob Langer about different opportunities that I had. So you have to pick Hopkins, and it's just been amazing, since day one, and I feel so honored, and so I'm very happy to share with everyone today some notes about my journey and to talk about all the different people that I owe so much thanks, and I feel so much gratitude to be here today. Just to start some disclosures, I am affiliated with some companies I'm going to talk about, and this is the translation, and I'll talk about that some more. Any potential conflicts of interest are managed by Johns Hopkins. So, so first to the Seder family, and we heard, we've heard about the tremendous impact that they've had to our institution and their love for Hopkins, and then also their vision and their generosity and what that can enable and into the future, into future generations. And this is very special for me, especially because, as I said, when I first came to Hopkins, I initially felt like an imposter, like I don't belong here. And I remember, faculty meetings, there were these huge figures in the field, and then more broadly at Hopkins, figures like Henry Brem, who I would hear, Bob tell these stories about, of how challenging it was for them to invent as an engineer and a clinician, and they'd submit grant after grant, and it's going to fail because of this, and then because of this, and because of this, but that perseverance in believing that you could create something new, and then that's what it takes, and then you impact the patients. But I remember then that there were these, even senior, more senior and experienced team that helped our department, the advisory board. And as a young assistant professor, I really felt sort of intimidated. But then one of them, with a very open heart, talked to me about how he was excited about the work I was doing on the cellular level and molecular level, and how this is important for BME, and it's great that they hired me. And so this was Bob Seder talking to me as a brand new assistant professor that joined the faculty. And then, in different years, connecting with him. And so it's really such a wonderful honor and pleasure to be the Herschel Seder Professor. And I, will try very hard to live up to all of the same ideals that he lived by, and his family, and I feel so proud and honored to be the third holder of this wonderful chair. And then, another wonderful thing with Bob was that in this very room, not too long ago, he gave a very inspirational lecture, and in part of it was the ideals of Hopkins and the mission and what it means for the world. And so the words that are hidden right there, you can see up here of our simple aim is to make scholars strong, bright, useful, and true, and about the impact that then Hopkins is going to have on the world. And then Bob also then proceeded to give these keys to success for an academic career, which actually I wish I would have had when I first started, but over 15 years, I started just a few months before you, President Daniels, I found that really each of these, it is the list to live by. With hard work, perseverance, passion, integrity, it just opens up doors. And then being in an environment like we have here at Hopkins with these amazing colleagues and having a little bit of luck too, you just can create wonderful things. And so I have so much gratitude. And so thank you so much for being so welcoming from day one, and for all your generosity to the Biomedical Engineering Department, and for you and your family, all your generosity to Hopkins. It's so well appreciated. And so now I'd like to talk a little bit about research collaborations and translation and some of the work my lab does. And a central challenge when I started here at Hopkins 15 years ago was this challenge of delivery of molecules that would then have intracellular delivery. And so the idea of drug delivery in a broad sense of having controlled release in the extracellular environment, this has been around a long time. There's a lot of solutions to it. There's a lot of targets that are extracellular. But what about if you want to deliver a cargo that's very sensitive and degrades easily, and you want to get it intracellular into target cells? Well, then it's very challenging. And so the approaches that are out there are viruses, and those can be very efficient, but they have many limitations in terms of safety and manufacturing and cargo capacity. And then some newer innovations now with lipid nanoparticles solve some of those, but it still has challenges with their safety and therapeutic window and the ability to target. And then other materials that are in the pipeline, like polymers, a lot of them aren't very good for gene delivery, intracellular delivery. They can be toxic, they're not very efficient. And so our approach was, well, let's make some new types of polymers, and if we don't know exactly how to make it when we start, let's make a lot of them, and then think of ways that we can screen them in a high throughput, high content way to learn then about the structure function, and then we can iterate on that and design new materials to do different things, to deliver different kinds of payloads, different kinds of cells, to then look at treating different diseases with vision, in brain cancer, and in many other areas. And so that's what we do. We design these biodegradable particles, and they're made out of polymers that are positively charged, and then the nucleic acids they contain are negatively charged, and those two come together like a magnet, and they self-assemble into these little nanoparticles that we design to be degradable. And so they have this general structure where we can then modulate the pieces that make up the polymers. And so we have, for example, certain parts of the structures that have those positive charges that let the cargo get protected and bound into this particle, and then we've looked at how we can modify the ends of the polymer to then have the particles go into some cells over others, cancer cells but not healthy cells, or stem cells and not non-stem cells, or a certain type of immune cell, and so on. Then a really big challenge with intracellular delivery is that you can get nucleic acids and particles in the cells, but then the cells, they eat those particles and they digest them and then spit them out, and like how we eat food. And so what we had to do was design groups in the polymers that could then buffer that endosomal environment, protect that cargo, and get it safely out of the endosome to the cytosol. And then the last component is that we make these materials from the bottom up so that they degrade very safely. And so they degrade in water in just hours to days, and we have even other groups that we put in there so that they can break apart in just minutes. And so this solves a big challenge in the field where a lot of these materials would cause toxicity or issues if they'd hang around too long. And so with those different building blocks, we then made different libraries of materials where we can change those building blocks that make up the polymers or the end groups or later branching to then see how we can understand more about the polymer structure and how it affects function with cells, and how we can then find new materials that can now open up new possibilities with medicine. And so what we found is that these materials, we can make them to be very safe and effective, where they can now function very much like viruses, but again with these biodegradable materials that are very safe and flexible. And we've validated this now in different kinds of applications, such as in the eye, that we can have a single injection and have very good penetration and distribution throughout the whole eye. So this is actually data from pig eyes together with our longtime collaborator, Peter Campachero, and I'll talk a little bit about that in a few slides. Other work where we can have systemic injection of particles, they can go to certain organs such as the lung, and this is an example with RNA delivery, and we're doing other work with editing and cystic fibrosis in the lung. Or we can have delivery in the brain, and this is a injection in the brain where we're seeing gene editing happening in the brain from our particles that can specifically do that. And so one of the challenges is specificity, that we'd want to deliver the medicines, but have it go to precise locations. So with lipid nanoparticles, they work well with COVID vaccines and IM injections, and they work well for a systemic injection to go to the liver. But if you want them to go to other targets, how can you do this? And so what we did is, by using these new materials that we innovated in the lab with the wonderful students in the lab, is that we could have materials that could go to the liver and express there, but they could also instead go to the lungs or go to the spleen or go all throughout the animal. And so we were really interested on the immune side about, well, could we get dendritic cells in the spleen transfected? Because the dendritic cells are like the conductor of the immune orchestra. And if we could specifically transfect them, we can have not just a strong antibody response, but we could have a very strong cell-mediated response. And this could make an even better vaccine for infectious diseases or open up new types of vaccines like for cancer. And so that's what we did. And this work was in collaboration with Drew Parole. And what we saw was that here's some animals that had colorectal cancer, and we injected our particles systemically. They went to the spleen, transfected those dendritic cells. Those dendritic cells activated the right cytotoxic T cells that went through the body and then attacked the tumor. And in this case, we had about half of the animals, the tumor shrunk away, and they were long-term survivors. And then if we gave them new tumors on their other side, those tumors shrank right away too, so that they were protected from that cancer, in a durable way. And then we looked at the cells in the blood, and we found that more than half of the T cells were T cells that were specific against that tumor. So there was this long-term response that then gave protection against that cancer. Now, in this case, we knew what the cancer was ahead of time. When some other work that we're doing is for the case, well, what if we don't know the antigen profile of the cancer ahead of time? Could we make something? And this is also something that goes back to Bob's work at the NIH with vaccines, is that when you develop a new technology, and this is something we're very mindful in the lab, we really want it to be something that can be affordable and accessible. And so if engineering cells can make great opportunities, if you have to take the cells out of the body, engineer them, it costs $400,000, and you put it back in, that's not going to help everyone in the world. But if instead, if you could have an off-the-shelf particle that you inject, and it engineers the cells in the body endogenously right there, and can have a very similar effect, now that's something that you could transform the world with and help those that need that medicine. And so the idea here is that we have an injection into a tumor, and then we reprogram the tumor. So we kind of flip it from being a bad guy to being an informant of the immune system, to then teach immune cells how to recognize it and other cancer cells that share characteristics with it. And so it's these reprogramming nanoparticles to reprogram the tumor immune microenvironment and to make a cancer cell then act like an antigen-presenting cell. And this is work that's done in collaboration with a very talented assistant research professor, Stephanie Zhang. And what we developed was particles that could transfect cancer cells very well, and that it could selectively transfect cancer over healthy. And then we delivered instructions then that reprogrammed the cancer cells to act like these antigen-presenting cells. And then what we saw was that we could have an animal like a mouse that has cancer in its flank, and then it also has cancer that's all through its lungs. And we could treat just the flank, and then the immune response will shrink that tumor, but those immune cells will go through the body and find the cancer in the lung and kill those too. And that's what you can see here. So this is in a control case that the tumors are all growing in the lungs, but then when we treat in the flank, those animals that have tumors in their lungs too, the tumors all go away from the lungs because that immune response is systemic, and you've activated now that cellular immune response with an off-the-shelf particle that was just injected. And then we see CD8 cells infiltrating into the tumors in both the flank and the lungs. So with research also comes translation. And so this is something that, again, I feel so fortunate from day one to have such wonderful mentors, including Sasha Poppel and Peter Campochero, they're both over there, who we were very interested in working together on new treatments for blindness, and especially a leading cause of blindness called age-related macular degeneration. And what we developed was peptide therapy that can be then sustained over time, because patients right now are treated where they need injections, in some cases even every month, intravitreally. And so by looking at new biological mechanism and then having the sustained release, could we have a new kind of treatment to really help them? And this affected me personally. My wife's mother would come to Wilmer to get injections with anti-VEGF from Neil Bressler, and it really helped her, but she was coming from Connecticut down to Wilmer because she really wanted to go to the best. She had a a condition that was rare, and then maybe also see the grandkids. But I saw how it affected her, to have an injection every month. And so Sasha and I, we sort of bootstrapped this because we were interested in the translation, but we didn't know how. This is before JHTV. There was some new incubator space that was soon to open up in Steep Silver, but nothing existed. We didn't have, we didn't have the vision yet with Ron and Christy and everything we now have with entrepreneurship. But we made it work with different kinds of grants, and then with Peter's leadership in all the models and in the disease, that together we grew Asclepics Therapeutics, and now you can see in the bottom over here a lot of the team, and it's led by Bob Dempsey up in Boston, and we're in the clinic and we're treating patients with the very same molecules that we were experimenting on the lab bench, a few years before. And then, from there's been some others. So with Dome Therapeutics, the focus is on the brain. And, Henry, again, is such a wonderful mentor. When I came to Hopkins, he introduced me to other neurosurgeons that were interested in different approaches. And one of them was a young Alfredo Quinones Hinojosa, Dr. Q. And together we collaborated on different approaches to look at cell and gene therapy to treat brain cancer. And this is an example of some work we did collaboratively also with Henry and a wonderful team here to have stem cells that can secrete factors to treat brain cancer, and then we could administer those cells intranasally or even intravenously, and they would go through the body, cross the blood-brain barrier, and get into the brain and home right to the tumors. And this is how we're looking at being able to then bring these medicines to hard-to-reach frontiers like the brain. And then more recently, there's been Cove Therapeutics, and the focus here is on gene therapy in the eye for retinal conditions that are of a genetic nature that we could then replace a good gene to then take over the function of the bad gene and cure blindness in those that are born with these retinal disorders. And together with Peter again, we've been working on this, and we went from models in mice and rats to models in pigs, and now non-human primates, and it's really exciting to do this. And then the business team behind us is also Niran Shah, the CEO, and Bill Woodward, chairman, and it's been really fun to work together on these translational problems. And then most recently, OncoSwitch, which is then that technology that I just showed a few slides ago to reprogram tumors, together with Stephanie Zhang, and then Emmett Cunningham, another Hopkins alum that's come back and wants to help create new translation with other faculty members. So there's so many to thank, and I'd like to just go through a little bit of a story of what brought me to Hopkins and just thank all these wonderful mentors and collaborators and students along the way. And so my research started as an undergrad. So this is me actually in Pittsburgh, at University of Pittsburgh. This is before you could afford to print out a whole poster. So it was $100 just to print out the banner, and then everything else I just had to print on the little board. But I had an opportunity to be an REU student after my freshman summer at University of Pittsburgh, and this was with Eric Beckman in chemical engineering, and I was doing green chemistry. I was using supercritical CO2 as a solvent to do epoxidation reactions as a chemical engineer, and I liked that a lot. I really liked research. But then as I went through the program, I really wanted to make an impact on human health, and so that brought me to the double major in BME, and then that became my focus. And I had a wonderful advisor for my senior undergrad thesis, Todd Probitzian, where I worked on using polyethylamine for DNA delivery, which is still what I'm doing today. So it really started with undergraduate research that I just had a love of, could we use these new emerging technology tools as engineers to then impact medicine, and that genetic medicine was something that was gonna come in our lifetime. And after COVID, there's been more than 10 billion injections of mRNA nanoparticles, we know this is here to stay, and that translation's all happening. So then I graduated Carnegie Mellon. Here's me with my parents, and then me with my future wife, Steph. We met in calculus class when we were 18, freshman year. And we've been together ever since. Yeah. And then I went to MIT, and at MIT, I had these wonderful mentors, Bob Langer and Dan Anderson. In those days, I'd come to come to work in Hawaiian shirts. I loved that's how I would roll. And I also had wonderful mentors on my thesis committee and as instructors, Doug Laufenberger and John Essigman. And so it was such a wonderful environment to learn, and I would learn like a sponge. And then what was it like a day in life in the Langer lab? Well, I found some old pictures. So we're being a little silly here, but just like these, looking up at old black and white pictures of professors in the lab, like maybe in Newton's time, having a pipe and being thoughtful about the science and then putting it into practice. But it was also something that we worked hard, but we also, the community and the friendship and enjoying being in lab and enjoying, the science. It was a very big lab. You could see Bob over here, and then here's Steph and me over here. And then, in a few years, it was time for graduation. And so here's graduation. Doug Laufenberger was the chair of my thesis committee, and then with Bob here. And this was very soon after he first became institute professor. And so I came to Hopkins. And I just been so blessed from day one to have such amazing mentors that then taught me to be a better scientist, a better engineer, a better human being, and to go on these journeys together. And so, Jennifer Alicieff and Henry Brem from day one, and then a wonderful series of directors in BME, starting with Elliot McVeigh, and then Les Tung and Mike Miller, and I've learned so much from each of them. They've transformed me in so many ways, and then so many others. So I mentioned Dr. Q earlier, John Schneck, who's here. That together we, as an immunologist and a bioengineer, this is more than a dozen years ago that we first thought, well, let's get together and create what now is immunoengineering, and now we direct together the first NIH-funded center for immunoengineering. It's the Johns Hopkins Translational Immunoengineering Center here, and it all started from, again, these interactions between engineers and clinicians working together on fantastic problems, and so too with Marty, with John Laterra, Denny Wirtz, and then Sasha and Peter, and then Hai Xuan from Material Science and IMBT, that we've had such wonderful collaborations together on gene delivery materials and new approaches to genetic medicine. And then with Henry, not just the collaboration with his lab, but opening up his doors to all the other neurosurgery faculty too, which I've enjoyed working with so much, and then through the time. But there's so many more people. So in bioengineering, I wouldn't have been able to do anything on that list that everyone is so kind to with the words about if it wasn't for all these other faculty and BME, both the primary BME faculty and joint BME faculty that working together on the research, on education initiatives, on translation, that has just been so transformative. And then beyond the BME, the other clinical collaborators, which aren't shy with the idea of a biomedical engineer entering the clinical realm, maybe even going into the OR, and then tackling these problems together and letting my students and trainees see firsthand what the clinical needs are, and then meeting together every 2 weeks for 10, 15 years. A few fellows from their lab, a few students from my lab on these interdisciplinary teams to solve these problems. And then we talked about translation. The reason that happens is there's this huge team behind that at JHTV and also in the ecosystem that want to see that happen and that work with faculty and support them with writing those patents, licensing those patents, having these opportunities. And I'm so grateful for all of them. And then all of the other staff and others to thank that have enabled the lab to run, the department to run, and all of these discoveries to be made and this innovation to happen. So it really takes a village. It's not something that's me. It's this whole team all working together because we have these common Hopkins values of trying to make an impact and translate and heal the world. And so the BME department is fabulous faculty and students, and here's just some some pics from that. We have these different events where we faculty and undergrads get together at the winter social, the senior send-off, and it's just, such a pleasure to work with different colleagues at conferences and also have some time just to have fun if there's a gala or if there's an extra day to go to the beach. And so this is what the Green Lab was when I started. I would work in the lab there. I was pipetting. It was Nipora and Ron as my first two grad students, and then Joel as a MD student that wanted to do a little research on the side. And then we had two undergrads, Marie and Monoe, that joined us. And then each of my grad students, I didn't know at first, but they all turned into superheroes. And they did the most amazing things that would impress me. And it was something that any kind of challenge, they would overcome it. And it's just been so wonderful. And so to just go very quickly through each of these superheroes. So Nipora Bise, who developed the first gene delivery polymers in our lab and was able to understand new mechanisms with particles per plasmids per particle and ways to use them for regenerative medicine and for induced pluripotent stem cells. And then Dr. Sunshine, Iron Man, who this very first immunoengineering work of how we can change the particle size and shape will change how it interacts with immune cells, and that we could program immune cells by making artificial cells out of particles. And this was a very big breakthrough in the field. We got a very strong patent together from it as well. And Dr. Sunshine went on to become a Siebel Scholar and is now faculty in dermatology and BME here at Hopkins. And Steph Zhang, who had more than a dozen papers, but here's one where what she innovated was new types of polymers that can deliver oligos like siRNA and microRNA, and it was making these new polymers that are bioreducible that was the key, and that they could also be very selective for targeting cancer over healthy cells. And she's faculty now at Hopkins as well. And so this was the lab at the beginning. We'd have more children than scientists. So it was like half, play with the babies when we'd have lab get-togethers. And here's Joel and Steph graduating, again, now both faculty. And after that, Ron Schumeli, who looked at vision and ways that we could have controlled release in the eye to extend therapies for longer periods of time. Corey Bishop, who developed different computational approaches to gene delivery, as well as looking at structure-function relationships through modeling and developing gold nanoparticle technologies in our lab. Both of them were just back for the 60th reunion. It was so nice to see them. Kristen Kozilski, who innovated a lot of work in brain cancer with reprogramming brain tumors to turn them off. She's now a professor at the Technical University of Munich in Germany. And so now our lab was really growing. We were a full superheroes team. The Ravens won that year. And then, we continued to grow. And each of these students, I'm just so thankful for their everything, their creativity, their perseverance, their friendship. Dr. Jae-Young Kim, who developed different kinds of pegylated particles for treatments in the eye, for cancer, for gene delivery, and he's now faculty now as well in Texas. Randall Meyer, who is MacGyver in a lot of ways. He could do anything. It could be an electrical engineering, a mechanical engineering. So he designed, for example, a new type of device to automate stretching of films to make nanoparticles of different sizes and shapes to modulate the immune system. And then other types of biomimetic particles in many other ways. He now is at Moderna, and here's the lab getting together. Dr. Wilson, who developed new types of vaccines, including self-amplifying mRNA delivery, and also looking at cyclic dinucleotide delivery to treat cancer. Camilla Zamboni, who looked at targeting of liver cancer over healthy cells, and then went on to become a wonderful physician. And our team of superhero mutants keeps growing. Yan Rei, who pioneered several approaches with ways that we can do CRISPR gene editing, as well as new approaches for mRNA delivery to hit other types of targets. And then Dr. Ray and Dr. Wilson ended up getting married, which is wonderful also. We had Mazir Mohammadi, who focused on the eye in ways that we can deliver different kinds of factors in parallel from gels that would then self-assemble once they're injected into the body. Hannah Vaughn, who looked at being able to have nanoparticles that we could inject systemically that would target just cancer and spare the healthy liver, and that could be used for both therapy and diagnostics, so to create these genetically encoded theranostics. Dr. Kelly Rhodes, who developed new treatments for autoimmune disease. So for example, to treat multiple sclerosis. So in this model, these animals develop multiple sclerosis. With these clinical scores, their hind legs stop working, they can't even walk. And then they're treated with the nanoparticles and would reverse the condition, that their scores went down to 0. They could go from being paralyzed to now walking because it turned on all the regulatory T cells that could protect myelin from being destroyed. And so here we are in our lab in the Smith building, overlooking the main hospital there. And then Dr. Ben Akiva, who developed the mRNA vaccines from a few slides ago, as well as artificial blood cells that could soak up toxins as a way to purify the blood. Johan Carlson, who developed particles we can inject systemically, and they could get even into the brain and cross the blood-brain barrier, and he's now faculty in Sweden. And Savannah Switty, who developed new kinds of biomimetic particles for immunomodulation, understanding how elastic modulus is key to affect immune cell interaction. And she's now at JHU APL, so she's still in the family. And Dr. Connor Hull, who then looked at the translational implications with brain cancer and how we can then scale up these formulations 1,000-fold and still have them work really well. And so to all these students, I just want to thank you so much for everything you taught me and for the journey that we've had together. And now we have the new generation of students in the lab, and it continues to be undergrads that do research. So Joanne Harris was an undergrad from Coppin that then did research in our lab and now just got accepted to a PhD program at Hopkins Medicine. I'll be coming here as a PhD student. We had a high school student like Nadia who did research with us over the summer and just got accepted to Hopkins BME as an undergrad. And so just like how when I first started as an undergrad doing research, it gives me such pleasure to help pay it forward with the next generation of students. And this is our lab today. A lot of them are here today. I'm so thankful to all of them for all of their absolutely fabulous work that they do each day in lab, and they're so collaborative with each other. Science has brought me to a lot of different places, but I love the lab so much and all the people in the lab and the culture of the lab. And then science also brought me to meet other people. I was able to meet heroes that I never thought I would have the opportunity to meet due to science, including people like President Obama with the P. Case Award, or people like Jimmy Page. So it's been phenomenal. And I'd like to thank especially also my family who's been with me, my parents that we didn't have as much money growing up, but they would make extra money set aside so in the summer I could go to the Museum of Science for summer camp for a few weeks, or we'd have a membership there because I'd always want to go and learn more about science. No one else in my family is a scientist. I was kind of the weird one out with that, but that really was transformative to me. And then, for my family, my wife Stephanie and my children, Kaya, Sid, and Luna, for their unwavering love and support. And then for all of you for sharing this with me today, thank you so very much.
Jordan, thank you, and I think it's pretty obvious why you are so deserving of this chair, and truly the world-changing work that you have done. As a sort of souvenir of this event from your installation, please offer you this, but let's go up in front. That's for you. Congratulations again. Thank you again, President Daniels. Thank you again, Bob Seder, Ted Luis, Mike Miller, and Henry Graham. And congratulations again to your entire family. Thank you all for joining us, and I invite you to join us for a reception.